Dear ladies and gentlemen, good evening. A warm welcome to our free webinar on emerging trends in bioinformatics, CAD, and machine learning. Today is a unique opportunity for each of you to unlock the treasure in the dynamic realm of bioinformatics. We are honored to have with us two exceptionally talented experts in the field of bioinformatics, Dr. Ashish and Dr. Om Prakash. We are confident that by the end of this session, you will fulfill with the valuable knowledge that will stay with you. It is my distinct pleasure to introduce our first speaker for today's webinar, Dr. Ashish. He is recognized with the M.K. Bhan Young Research Fellowship and securing the second prize in COBLET 2022. He is specialized in NASH-related molecular processes and protein conformational changes, and in many more in the field of bioinformatics. So, without further ado, let's welcome Dr. Ashish.
Yeah. Thank you for the kind introduction. So some of them are texting that there is no voice. Am I audible?
So you're audible.
Yeah, thank you. So I'll just share my screen. Is my screen visible?
Yes, sir.
So hello everyone and warm welcome to our webinar. So whether it's morning, afternoon or evening for you, I'm delighted that we have gathered here from different parts of the world. So today my presentation topic is unveiling the power of CAD in drug discovery and its innovation and success stories. So before starting my presentation, I would like to inform that I have some throat infection which might irritate or disturb some of the participants. So I kindly ask for your patience and understanding for the same. Thank you. So the agenda of my talk will be, I'll start with the basics of the biological macromolecules and its significance in CAD, an introduction to the CAD and its significance in drug discovery, key techniques and methods in CAD. And then I'll take some case studies illustrating successful drug discovery and using CAD. Then I'll be taking some case studies from my own work. And finally, in last, we will have some question and answer sessions. So first, I'm going to talk about the biological macromolecules, which are the protein, DNA, RNA, carbohydrate, and lipids. So these biological macromolecules are the building blocks and make up every living things on the earth. So here, I mean every living things means every living thing, whether it is the tiniest bacteria or is the gigantic blue whale or you as yourself. So what if these biological macromolecules aren't quite right? So things can get tricky. So think of it like a puzzle. So if a piece is missing in the puzzle or doesn't fit, so the whole picture gets changed. So in our bodies, if these important molecules get messed up or changed, it can lead to health problems. So for example, if the instructions in our DNA get mixed up, it might cause disease like cancer. Or if your protein doesn't fold the way it folds, so it can lead to conditions like Alzheimer's. So these tiny changes in building blocks can really affect how our bodies work. So to understand how these molecules work together and how alterations in it leads to the disease condition, scientists need to unravel their 3D structures. So once we have the 3D structure, we can understand the mechanism And once we understand the mechanism, we can design the drug molecule to treat or alleviate the disease conditions. The traditional methods of doing this drug discovery are expensive and time consuming. So to speed up the process and gain the insights into these molecules, scientists have turned to the computer-aided drug discovery. So in order to speed up and find a new lead molecule, the CAD approach uses the computational techniques, algorithms, and softwares. So imagine your target as a lock, and there are millions of keys to try out and find the right one to unlock it. For example, if key fits to the lock, then we can say it as a hit. In case of drug discovery, we see it as hit. What does it mean? It means that the molecule has the ability to bind to the target binding site. And if we are saying it as a lead, it means it also has the therapeutic effect. So the hit is the compound which has the binding ability to the target, but the lead molecule has the binding ability as well as it has the therapeutic effect. So, for example, it key opens the lock, means we had the desired effect that we wanted. We wanted to open the lock, it opens. So it will be considered as lead, whether we want to lock or unlock. In case of drug discovery, whether we want to activate some target or inactivate the target. So if the compound is able to activate or inactivate, has the therapeutic effect, then it will be considered as lead. And from lead to for the development of drug, it again needs the it again requires the further optimization process. In the optimization, the optimization of the lead will improve its efficacy, potency, bioavailability, etc. So what are the computational techniques that we used in drug development? So in the drug development process actually involves the target identification, target validation, lead discovery, lead optimization, and drug development. So for the target identification, scientists use the bioinformatics approach, inverse docking, inverse QSR, and once they have the target identified, they go for the binding site prediction and cavity searching inside the target. And once they have the binding site predicted, they perform the docking and library screening, and after that they perform the molecular dynamic simulation. And for the lead optimization, they use the QSR and pharmacophore approach or in vitro ADME approach. And once they have the lead optimized, they go for the preclinical testing and clinical testing for the final drug molecule development. The traditional method of drug discovery approach usually takes 12 to 15 years with at a cost of more than 1 billion. To reduce this time and cost, the scientist is using the CAD approach. So CAD approach actually speed up the drug development process in the early stage of the drug discovery. So in the traditional approach, you can see that the scientists use the target identification and screening, experimental screening, and through the experimental screening, they get the candidate compounds. So once they get this candidate compound, they go for the optimization using the human-led cycle, and they keep it optimized in multiple cycles. They synthesize and optimize it in multiple cycles, which actually takes a very huge time. To reduce this time, the scientists started using the in silico modeling, which is actually fast as well as it's accurate as well. So in silico, likewise, in vitro testing, in silico modeling must also follow the in vitro testing and in vivo modeling before the drug reaches to the market. So computer-aided drug discovery is subdivided into two fields: the structure-based drug design and the ligand-based drug design. So I'm not the expert of the ligand-based drug design, but I'll tell you the basic concept, how they use this ligand-based drug design. And in case of structure-based drug design, they first start with the identification of the target, then they go for the binding site prediction, and they perform the molecular docking. And so for the molecular docking, they take the library of the compound and they perform the docking-based virtual screening. In case of virtual screening, what actually it does, it looks for the possible binding site inside the target. So from the library of compound, so the compound which actually fits in the pocket, we take that compound, and each compound will have the multiple poses in the binding site. So and each pose will get a score from the scoring function of the docking tools. So here you can see in this animation, you can see that the ligand is taking the multiple pose, and each pose will be scored. So the pose which will fit in the, like, which will have the best fit in the pocket will have the highest score, and the highest score compound will be selected for the further optimization and experimental validation. And in ligand-based drug design, we use the pharmacophoric approach and the QSR approach. The pharmacophore is like a molecular map. It highlights the essential features in a molecule which is necessary for to interact with the biological targets. So, and in QSR, it is a mathematical and it is a statistical and mathematical model which actually links the chemical structures of the compounds to the biological activity. So what is the difference between the pharmacophore and QSR? QSR is the one which does not actually predict the biological activity. It will just highlight the essential features, the structural and chemical features of the molecule, which actually required for binding with the target. But QSR is the model which actually links your structure with the biological activity. So when do we perform the ligand-based drug discovery? For the ligand-based drug discovery, you should have the ligand information present, and if the target is unknown, even though if the target is known, the structure is not available. And if the target structure is even available, it is but the target is targeting the target is very challenging. And if you do not get a satisfying result with the structure-based approach. So as I was discussing that for pharmacophore modeling, we start with studying a known active molecule and their interaction with the target protein. The critical component of the pharmacophores are hydrogen bonding. Hydrogen bonding means it can be either hydrogen bond acceptor or the hydrogen bond donor. And it will be the electrostatic component like anionic and cationic component, or it can be hydrophobic component like aromatic or hydrophobic component. So in this case, it doesn't care of the which type of atom a molecule have, whether it is oxygen or it is a nitrogen, it doesn't care. It just take care of whether it is a hydrogen bond acceptor or donor. So oxygen can be hydrogen bond donor or acceptor, or the nitrogen can be also the hydrogen bond acceptor or donor. So it will just assign the feature whether it is a hydrogen bond acceptor or donor. So likewise, it determines the features of a molecule which actually involved in the binding with the target. So once we have the pharmacophore model, we will utilize it for screening. We will use this pharmacophore model for screening a large database to find a similar compound which have the similar biological effect. And in case of QSR, like you have the list of the compounds with the known biological activity, and you use to build the QSR model. So using this known biological activity, you model the QSR model, and once you have your QSR model built in hand, then you provide a molecule with unknown activity and you predict the its property. So what kind of the property which actually QSR model predicts? So these are the some list of the property like biological activity. It can predict the biological activity. It can even predict the toxicity. It can predict the solubility. It can predict the permeability, bioavailability, partition coefficient, chemical reactivity, metabolic stability, binding affinity, and mutagenicity. So based on the activity that you are providing, so it will predict the property of the molecule. So QSR model is actually built on based on the machine learning techniques. So the next speaker, Dr. OP, will be, I think, giving more insight into the machine learning techniques. So you'll understand how actually the QSR models are built. So now coming to the structure-based drug design. So when do we perform the structure-based drug design? The target must be available, and if you want to find out the interaction between the target and your small molecule, or you want to find the interaction between the protein and protein, and if the target is actually complex, and if you have a challenging ligand feature, which for that you are not able to design a pharmacophoric feature or QSR model, and in case your ligand-based drug discovery get failed, and if you want to disturb the protein-protein interactions. So target identification in structure-based drug discovery, the target identification is a crucial phase in drug discovery. So it involves for the specific molecule that can be modulated to achieve a therapeutic effect against a particular disease. So what should be the criteria for the good target? It should be the safe, it should be the efficacious, it should have a clinical relevance, and it should be the commercially viable. So what do you mean by safe and it should be efficacious? So safe means your target should have a therapeutic effect, but without having any side effect. It should not have any adverse effect. And what do you mean by here clinical relevance? So it should have the capacity to modulate the target and align it with the clinical benefits for the patient. And here the commercial viability means target should align with the market demand and have a potential to attract the investment from the pharmaceutical companies. Like when your target could be commercially viable, so it should have a factors like the disease prevalence, the patient populations, and potential market size will influence the commercial viability. So what are the approaches for the target identification? So you can either go for the literature survey, you can do the peer-reviewed article, you can review the peer-review article, you can go and find look into the journals and patents, and either you can look into the database like PubMed, GenBank, and DrugBank, or there are other approaches like you can do the gene expression studies, cell-based assays, genomics, transcriptomics, and proteomics, and metabolomics. So these are the approaches which actually helps in the identification of the target. So if your project is about a target-based drug discovery or a structure-based drug discovery project, so what you need to do first, you will need to have several 3D structures, one or several 3D structures. So you have to first check whether the structure is available in the PDB database. So whether the experimental structure is available, the experimental structure will come from either X-ray or NMR or cryo-EM. If the structure is available, then you can proceed with it. Otherwise, if the structure is not available, then you have to predict the 3D structure from the amino acid sequence. So for the prediction, you can either take the approach of the homology modeling, like you can use the modular server, or you can take the structures from the AlphaFold or the Rosetta Fold, or there are other related tools for the modeling of the protein. So next, assuming that you have your target molecule in 3D and with its binding pocket known, so and you want a small chemical compound which fits to the binding, which fits in the binding pocket of your target protein. So what you will do, you will screen a library of the compound or the database, and you will perform the virtual screening, the docking-based virtual screening, and then again you will get you will, like here you can see that after the docking, the compound has bound at the two sides, but you can select the base docking pose based on its ranking score. But even though if you want to check whether these compounds are stable inside the pocket or not, then you have to perform the molecular dynamic simulation. And not only the stability, if you want to check the how this compound is affecting the protein conformation, that also you can check using the molecular dynamic simulations. So once you get to know that the target is stable and it is able to change the conformation in either inactive state or the active state. Then you can go for the further validation of your compound to check its biological effect, biological activity. But sometimes you do not able to find the binding pocket inside the protein. So in that condition, you have to either use the target prediction tool, the pocket prediction tools, and there are several pocket prediction tools available online. And even sometimes the pocket prediction tools is also unable to identify the pocket site. So in that case, it is a cryptic pocket. So in that case, what you have to do, you have to either you have to either perform the simulations, so which will actually make the conformational changes to find out the pocket inside the protein, and either you can perform the fragment docking or search for the hotspots. So these are the some softwares which is used for the docking purposes, like Autodock Vina, Gold, MOE, and Schrodinger suits. So among all the Schrodinger, the Autodock Vina is open source. Open source here means it is freely available, and Gold, MOE, and Schrodinger is commercially available. Autodock Vina actually used the semi-empirical scoring functions for scoring the interaction, for scoring the which particular pose is fitting best to the binding pocket. So here semi-empirical means it will use both. It will combine both theoretical as well as the experimental parameters to evaluate the binding affinity inside the pocket. And Gold uses the genetic algorithm for scoring. So the genetic algorithm here means it is inspired by the principles from the natural selection and genetics. And MOE and Schrodinger suits integrates various other tools for the drug discovery process, and it offers many advanced features for the drug discovery and development. So now coming to the emerging trends and technology in CAD. So when we explore the latest advancement in CAD, so one standout trend is integration of the machine learning approach. So here I'm you can see it is written G Nina, but the pronunciation of the tool is Nina. So we do not pronounce it G Nina, it is just Nina. So Nina is derived from the Autodock Vina. So it has it actually combines the molecular docking with the convolution neural network to enhance the accuracy and speed in the docking simulation. So this is the algorithm behind the Genina. So it actually during the docking process, it uses the Monte Carlo method for looking for the all possible conformation of the ligand inside the binding pocket. So it is an iterative process. You can see here it will do the fast refinement using this, and in the and even it will have the multiple Monte Carlo chains running in parallel. So it will exhaustively search all the possible conformation which is to look for, and once it will search for the after searching all the possible conformation, it will merge all the best poses from each run, and it will go for the full refinement. Here the full refinement means after the docking it will optimize the docking poses. So what do you mean by here the optimization? So you can think of you go to the market and buy a sugar, and after buying the sugar you fill it in a jar. So once you fill it into the jar, you some of the sugar are left in your packet. So what you do, you just shake your jar to just settle some of the sugar that were on the upper layer. So likewise in the refinement also, what convolution neural network will do, it will also it will just shake your molecule a little bit to adjust your molecule inside the pocket. So after the full refinement, it will rescore and sort based on the this Shannon score, and then it will do the RMFD filtering. So what does it mean here? The RMFD filtering means the most close posage, like which will be very similar, will be taken out from the study, and finally we will have the best pose from the Nina tools. So these are the some molecular dynamic simulation tools like GROMACS, EmberMD, NAMD, Schrodinger, OpenMM, and TorchMD. So among these tools, the GROMACS, NAMD, OpenMM, and TorchMD is freely available, and Ember and Schrodinger is a commercial tool. So, and TorchMD here is again a deep learning. It uses the deep learning framework for the molecular dynamic simulations. So it enables the molecular dynamic simulations calculations faster without compromising the accuracy. So this is the only tool which actually uses the machine learning approach to enhance the simulation process. Now coming to the application of molecular dynamic simulation. So what are the some applications here? You can perform the molecular dynamic simulation for the conformational exploration. So you have the starting structure predicted from either X-ray or you either like it is either experimentally solved or the predicted structure. So and you perform to get the simulation trajectory to see how to see its like how that it actually to see its dynamics and flexibility, and even you can perform the model refinement. So you have the starting starting protein structure which has the very high energy, and after performing the molecular dynamic simulation, you can see that your structure has been refined and it has attained a lower energy. So here also you can see that in lower energy conformation, you have a regular secondary structure formed which were actually missing in the previous high energy model. So molecular dynamics simulation also helps in refining the protein structure, and sometimes the loop refinement, like in the loop, sometimes you have the regular secondary structure, but it is absent in the initial structure. And here you can see the anti-parallel beta sheet has been formed in the loop side. So, and you also some of the other applications to determine how a biomolecular system will respond to some perturbations. Like if you add or remove a ligand and perform the molecular dynamic simulations to see how it is actually impacting the conformation of the protein structure, and you can even model the you can add the point mutations to the structure and see how actually it is impacting the structure and function of the protein. You can even alter the protonation state and see the impact over the structure and function. You can even apply the mechanical forces to see the pulling of the ligand or putting the ligand into the binding pocket. So here you can see this simulation in the steered molecular dynamic simulation. You can see the pulling of the ligand or binding into the pocket, and you can even apply the external potential to see to see the movement of the ion into the transmembrane proteins, and even you can alter the membrane compositions. Here you can see the membrane compositions are somewhat different in than this one. So if the membrane composition is changing, then also the protein conformation is changed. So here you can see the previously the channel is closed, and after the changing the composition of the lipid, the channel is open. So that also you can check using the molecular dynamic simulations. So what are some other applications? You can even check the observe the dynamic process over time. You can check the transport of the ions across the membrane. You can even check the conformational change, like after adding and removing the ligand, how the conformation is being changed. So here you can see an allosteric effect. Allosteric effect means if you are making some changes at one site, and its effect is coming to some other side. Structural effect is coming to some other side. So I'm having a case study for this particular part in the example that I'll be showing. So you can even do the you can do the protein folding. You have initial structure which is completely unfolded. You can perform molecular dynamic simulations to check its fold, and sometimes even you can check the ligand binding and assembly pathway. So what are the some success stories? Here are some success stories of the drug discovery, and optimization through Cadd. So imatinib, which is marketed as Gleevec. So it was designed for treating the chronic myeloid leukemia, as well as GIST, which is gastrointestinal tumor. So it was designed based on the Cadd approach. It designed the designing was very high, it was very high, very highly selective molecule for the target. It usually targeted the BCR avial fusion protein, and it was first approved in the 2001. And oseltamivir, it is marketed as Tamiflu, which is designed for treating the influenza, and it targets the minidase. And actually, oseltamivir was first designed by the experimental techniques. It was screened by the experimental techniques and by the medicinal chemist, and later on it was optimized using the Cadd approach, so for improving its binding affinity. And it was first approved in 1999. And raltegravir and darunavir and dolutegravir, these three drugs were designed for treating the HIV. And raltegravir was first approved in 2007, and darunavir was approved in 2006, and dolutegravir was approved in 2013. And venetoclax is the drug which were designed for treating the chronic lymphocytic leukemia, and it was targeting the BCL2 protein, and it was first approved in 2016. So this list is actually very small. There are the list is actually very big. The drugs which has designed from the computer-aided drug discovery, I think it is, if you search, you can find more than 100 drugs which has been approved using the Cadd approach. So now coming to the case studies from my own research. So first, in the first case study, I'll be checking how GJB1 mutations impact the connexin 32 functions and Charcot-Marie tooth disease. So connexin 32 protein is encoded by GJB1 gene, and connexin 32 protein is an ion channel which actually forms between two cells. So in 2012, Abraham and their group reported that positive charge of the arginine residue at position 75 plays a very critical role in the normal functioning of the gap junction beta 1 protein. But the molecular mechanism, how this particular mutation is impacting the function of the connexin 32 was not known. So therefore, a thorough understanding of the impact of mutation at this particular position is very important. And so we perform the molecular dynamic simulations to understand the mechanism. So here you can see that the different color, the different color of this monomer represent the connexins. So in a single cell membrane, 6 connexin molecules of unit come together to form a connexon, which is actually a hemichannel. Hemichannel is present in the one cell membrane. And to form a full gap junction, the two hemichannel must dock together to form a gap junction ion channel. As Abraham has reported that these 3 mutation is actually impacting the function of this connexin 32 and leading to the Charcot-Marie tooth disease. So I perform the molecular dynamic simulations to check how this is actually impacting the connexin 32. So after the molecular dynamic simulation, what I found that I performed the network analysis. And in the network analysis, what I found that the mutant, the wild type where the arginine was present, in the network it is making 34 salt bridges, but all the three mutants is making just 33 salt bridges. The salt bridge, the additional salt bridge in the wild type was between the R75 and Q47, the glutamine. There was a salt bridge between the glutamine and arginine, which actually were missing in all the mutant form. And because of this like missing salt bridge, what I found, then we perform the like pore channel analysis. So here the green color means here the blue color means it has the capacity to it has the diameter to for the two water molecule with this, and in green color means it has the diameter for just single water molecule, and red color means it is very tight for the water molecule, so no water molecule can pass through the pore channel. So for this connection, the first one is the wild type, so and the other three is for the mutant. So in case of mutant, you can see that this pore channel has been like greatly reduced. You can see the more red colors in it. So because of this mutation, this pore channel has been completely shut. So that was the reason how it is impacting, how a single mutation is impacting the pore channel and leading to the disease condition. The conclusion was the mutation from R75 R75P mutation drastically reduced the pore dimension, and R75Q and R75W mutant completely shut the pore channel, which could alter the transport of the ions and small molecule across the nerve cells and hindering the communication between the nerve cells. And consequently, this may lead to the association of mutants to the X-linked Charcot-Marie tooth disease. Now coming to the case study 2. So this is my current work. I'm presently working on the non-alcoholic steatohepatitis. So non-alcoholic steatohepatitis is a more severe form of non-alcoholic fatty liver disease, which is the leading cause of the chronic liver disease in the United States. And NASH is the fastest growing cause of the liver cancer death globally. And NASH is now is a global public health problem in India as well, and its count is gradually increasing after type 2 diabetes mellitus. Until now, there is no appropriate drug to treat the NASH condition. So an urgent global effort is required to address the underlying metabolic risk factor and minimize the burden of liver cancer caused by NASH. So this is how this is the progression of the liver disease in case of normal liver. When in starting you have the normal liver, and when the fat start getting accumulated in your liver, this condition is called steatosis. And when more of the fat will get accumulated, then this condition will be called as NASH. Because if the more fat will get accumulated in your liver, then it will cause the inflammation in your liver, and which will lead to the death of some of the hepatocyte cells, which will start causing the scarring in your liver. And once the scar will be formed in your liver, then some of the fibers will come and replace that scar. And then at that condition, we say it as the fibrotic liver. And that condition is known as the cirrhosis or the cancer stage. Until now, there is no drug which can treat this condition or reverse this condition. And the only treatment is available is the transplant. And if the patient is unable to take the transplant, then unfortunately it will go for the death. So what are the pharmacological management that is has been taken approach? There are few companies which has designed the drug for there are few companies which are started designing drug for this particular disease, and they started designing the inhibitor for the ASK1 protein, because there are several studies which has demonstrated that ASK1 get overactivated in case of NAS condition. So Gilead is a US-based company which started with the selenosertib, and their drug worked very well till phase 1 and phase 2 clinical trial, but it got failed in the phase 3 clinical trial. And there is another company which has come up with the drug SRT-015, which is also a ASK1 inhibitor, and it is currently in phase 1 trial. So what is the reason actually the drug is getting failed in phase 3 clinical trial? So the main reason was the side effect. So it has a very huge side effect. So because of that, it got failed in the phase 3 clinical trial. So instead of, and another main reason was they were targeting the catalytic site of the ASK1. So if you are targeting the catalytic site of the protein, so it will even interfere with its normal physiological function as well. So instead of targeting the catalytic site, we are targeting the PPI site, which is protein-protein interaction site, which is an advanced method and has the potential, but it's challenging. So in case of NAS, here you can see that the ASK1 protein has the balanced PPI, but in case of NAS condition, this PPI interaction between the NAS has been imbalanced, which is causing the NAS in the liver. And the therapeutic approach previously that the other companies have taken is like the conventional approach. They were targeting the catalytic side, which actually interfere with the normal physiological function of the protein as well. But we want to modulate the condition. We don't want to completely suppress the ASK1. We want to just modulate its overactivity and bring down it to the normal level to maintain its normal physiological function. So using the PPI approach, our aim is to maintain its normal physiological function instead of completely suppressing it. So this is actually a very complicated diagram. So in this, I have first reviewed all the ASK1 PPI regulators. Like here, I have curated all the protein interact like all the protein which actually regulates the ASK1 protein. So here, whatever the arrow you are seeing in the red color, it actually leads to the inhibition of the ASK1 protein, and whatever the arrow you can see in the green color, it actually leads to the activation of the ASK1 protein. So our goal is to our aim is to inhibit the ASK1 protein. Then I will focus on the protein which actually negatively regulates the ASK1 protein, which suppress the activity of the ASK1 protein. So I started with the 1433 protein. The 1433 is the protein which actually suppress the ASK1 protein. But how does it suppress the ASK1 protein? The mechanism is not known. So we perform the microsecond molecular dynamic simulations to see how actually the 1433 suppresses the ASK1 protein. So, and the one more thing is to focus is like 1433 binds to the ASK1 protein to its phosphorylated serine residue. So so here I'll show like how. So how does actually the serine phosphorylation at ASK1 inhibit the kinase activity? So I wanted to understand whether the sorry, I think I missed one slide here. So we had the 1433 structure bind with the ASK1 peptide, and we had the ASK1 kinase domain bind with the selenosertib ligand, which was designed by the Gilead company. But there was a missing structure between these two. And if we want to understand the impact of the 1433 over the ASK1 catalytic site, so we have to model these structures. So instead of modeling this structure, we took the AlphaFold model and aligned it with the ASK1 peptide. So this is actually the AlphaFold model of the ASK1 protein, which I aligned with the 1433 protein, and I made a complex between of the 1433 and ASK1. So first I wanted to understand whether the phosphorylation is important for whether this phosphorylation is important for the interaction with the 1433 or not. So we perform the simulation with phosphorylation and without phosphorylation. So here you can see that when the peptide bound without phosphorylation, it was actually unstable at the pocket of the 1433. And when it had the phosphorylation in the serine residue, then the peptide was stable inside the pocket. So next, we had the complete model. We had the 1433 as well as the full ASK1 protein along with its catalytic site. So here I simulated, I performed the 1000 NS MD simulation and see that this particular domain, which actually the peptide domain which we saw previously in the crystal, it is completely stable inside the pocket. And then we wanted to see how actually this is impacting at the catalytic site. So here, this is the 1433 and ASK1 complex simulation and without selenofertib. Here the drug is just kept to see how the binding pocket is actually, how the conformation of the binding pocket is actually changing. So here you can see that when the 1433 binds with the ASK1 without any drug, it is actually closing the pocket of the ASK1 protein at the catalytic site. I even performed the molecular dynamic simulation without 1433. So here you can see that, sorry. So here you can see that when selenofertib drug was simulated without 1433, it was completely stable inside the pocket of the ASK1. But when the 1433 were bound with the ASK1, you can see that the drug become unstable and it is moving outside of the pocket. So I performed the catalytic pocket volume analysis of all the complexes. Here the ASK1 apoprotein means there is no ligand and it is without 1433. And this is the 1433 ASK1 domain without selenofertib. This was the 1433 ASK1 with selenofertib, and this is ASK1 selenofertib. In the pocket volume analysis, you can see that when ASK1 selenofertib, its pocket volume, the average pocket volume is 724, and when the 1433 added to the ASK1, then its pocket volume get increased and the average pocket volume is 784. And when there was no selenofertib present inside the pocket, its pocket got reduced to 436. The average pocket volume is 436. The final conclusion comes to 1433. How does 1433 actually regulate the ASK1? The 1433 negatively regulates the ASK1 by its allosteric mechanism. So in the absence of substrate, it reduces the ASK1 catalytic pocket volume to the extent that the substrate cannot enter. And in the presence of substrate, it increases the ASK1 catalytic pocket so that the substrates become unstable and moves out of the pocket. So these molecular insights seems pivotal for the mechanistic understanding and 1433 ASK1 interface specific therapeutic interventions. And from this 1433 ASK1 interface, we design 10 peptides, and these 10 peptides were experimentally we perform the BLI assay. BLI assay is a very powerful method to study the kinetics of the molecular interaction, particularly in the context of the protein and protein small molecule. So we perform the BLI assay for all the 10 peptides, and here we calculated the Kd value. So lower the Kd value is higher the affinity. So here you can see that the peptide 9 has the highest affinity with the ASC1 protein, and other peptides has like the rest other peptides are listed in the decreasing order. But here I wanted to stress that just binding doesn't determine like it will also have a therapeutic effect. So here you can see that the peptide 9 was having the highest binding affinity, but when I did the other assays to confirm whether it has the biological activity, we did not find the activity for the peptide 9. So here you can see we perform the flow cytometric analysis for the electrocell line. Electrocell line is the liver cell line. And we perform the annexin assay here to see the annexin assay is usually to check the apoptosis. So in case of like the first one is like without treatment, and other 4 are with the treatment of the peptides. So the without treatment, we saw that there was 23% of apoptosis, and with the treatment of the peptide, you can see that the apoptosis got reduced to 9%, 13.3%, 6.29%, and 10.3%. So in case of peptide 5, the apoptosis reduced to at least 4-fold. So among all the peptide 5 is having the highest therapeutic effect, and we are doing some other assays also to check the therapeutic effect of the peptides for the NASH disease. And thank you. And if you have any questions, you can. I believe I completed in the stipulated time. Do we have any questions from the participant side?
Probably the questions are being shared at the chat box also.
Okay. So Divya is asking, which tool did you use for the pocket volume analysis? I use the POVME tool. POVME, POVME.
There is a request for every participant. If you have any questions, please raise your hand so we can unmute yourself.
Yeah, Genie is free. Yeah, case studies were little complex to understand in one go. Yeah, I do agree. If you have any like question, and if you are working on any structure-based drug discovery project,
Hello, doctor.
Yeah.
Yes, I would like to know that your session was very excellent and engaging also. I want to know that you discuss about the Genie now.
Yeah.
Is it free software?
Yes, it's free.
And second, what is the difference between dynamics and simulation, protein dynamics and simulations?
We perform the simulations to check the protein dynamics. Simulation. Are you getting? Simulation is, why do you perform the simulation? Simulation is something to mimic the behavior of something. Simulation, meaning of the simulation is to mimic the behavior. So we want to mimic the behavior of the protein and ligand, how actually it behaves inside our body. So we perform the molecular dynamic simulations to mimic the behavior, how it is actually behaving inside the body, and we want to see it inside the computer by providing the environment that actually it is present inside our body. We cannot provide the actual environment, but we are trying to mimic the possible like environment to check the dynamics at its molecular level. Are you satisfied with the
Hello.
answer, or do you hello?
Hello. Thank you for your presentation. It's very excellent. But I have a question about the case one study. I wonder that the mutated structure came from computer prediction or came from the wet lab. And if they are predicted, if they were predicted, I'm not sure are they reliable for the conclusion.
Yeah, the predicted structure were actually were taken from, like, we perform the modeling for the predicted structure and for the reliability. And previously we were having like the modeler and some other tools for the modeling which were actually reliable. Like previously, if you were having just accuracy of 60 to 70%, then also we were picking and we were happy with the target structure. And now you know about the CASP, which is actually the critical assessment for the structure prediction. So in 2018, when the AlphaFold came for the first time, and before the AlphaFold came, all the previous tool were having the structure prediction accuracy were approx 60 to 70%. And when the AlphaFold came, it was actually having the prediction accuracy less than 50%. And when AlphaFold came, it has the prediction accuracy was approx 70%. And which were actually a great breakthrough at that time. And when the AlphaFold version 2 came, so it has the actual the prediction accuracy was approx at least 90%, which is, you can say, that equivalent to the experimental structure. So now if you are taking the structure, like if you are working on the structure-based drug discovery project, you can take either the AlphaFold structure which is available in the website. If you don't have the structure in the website present in the AlphaFold, so then you can either use the AlphaFold tool for modeling your structure from the sequence.
Okay, so after we predicted with AlphaFold tools, then we have to confirm the result with wet lab. Is that right?
Yeah, for any bioinformatics prediction, ultimately you have to go for the validation through in vitro and in vivo test.
So last question. When you have to like publish a paper, is that okay to have the wet lab to confirm the result?
It depends, like, which journal you are selecting. Like if you are selecting a journal which only takes the computational paper, then you don't have to show them the experimental validation. But if you are selecting a journal which actually takes both structural prediction as well as the experimental validation, so you have to do the validation to target that journal. And usually it is recommended that if you are doing any prediction, you validate it using the experiments.
Okay, thank you very much.
Yeah.
Hello, sir. Thank you for the informative lecture. I want to know, you told us that we can know about the targets by either databases or literatures. If we are going with the proteomics, metabolomics, and other things, does these all come in wet lab or dry lab?
It comes both. Like if you are going for the genomics, transcriptomics, and proteomics, first you have to start with the wet lab. Then you do the bioinformatics analysis, because you will be through the genomics, proteomics, and metabolomics data and transcriptomics data, you will be having the huge data. To analyze that data, you will be again you will be needing the pipeline to analyze all those data to check like how actually it is. Like for example, you have healthy case and you have the disease case. So if you are taking the genomics from a healthy and disease, then how you will compare? So if you are having the huge data, then how you will compare these two states? So again, you will be needing the bioinformatics pipeline.
Okay, sir. Thank you.
Is there any other questions?
Hello, Adabal.
Yeah.
Doctor, how can we perform the simulation?
How can you perform the simulation?
Yes. How can we perform the
Hello,
simulation?
I have shown you some of the tools, like you can use the GROMAX or Ember, or if you have the commercial software, then you can use like Desmond, which is provided by the Schrodinger.
Desmond?
Desmond.
Okay,
So
thank you.
if you are like if you are starting, so you can start with the free version of the tool, which is GROMAX, which is like widely used for molecular dynamic simulation.
Yes, doctor, thank you. And secondly, how we perform simulation before the modeling or after the modeling?
The prerequisite of performing the molecular dynamic simulation, you must have a protein structure.
Yes. So,
Yeah.
okay, sir, thank you.
Hello, sir. Sir,
Hello.
protein topology in GROMAX. What should we do if protein is in protein? Some residues are miss, and what software we use to remodel that protein during preparation of protein topology to replace in fixed place that missing amino acid residue in that protein.
Yeah, for the missing residue, that also depends on like how many residues are missing, whether it is like very a number of residue should be like known, like how many residue you want to model. And if it is a very big numbers, then you can choose the like homology techniques like modeler. You can